Hello?
Hello, Xavi.
Okay, just wanted to know that someone actually is hearing me. Good, so welcome to this cycle of webinars of Bioinformatics Barcelona. I'm going to make an introduction in Catalan, then we will move to English for the presentation. And for the questions, you can actually ask them in the language that you prefer, Catalan, Spanish, or English. Bé, benvinguts al cicle de webinars de Bioinformàtics Barcelona, que encetem avui amb un convidat ben especial, en David Torrents, professor de recerca i crea al Barcelona Supercomputing Center i vicepresident de Bioinformàtics Barcelona. Com tots sabeu, quan al BIP parlem de bioinformàtica, ho fem en el sentit més ampli de la paraula, és a dir, l'aplicació i el desenvolupament de mètodes computacionals en biologia. Això inclou coses tan variades com la genòmica, el descobriment de fàrmacs, l'estudi de l'estructura i la dinàmica de sistemes biològics en diverses escales d'espai i temps, el tractament de dades mèdiques i el seu ús per la prescripció, per la predicció personalitzada, les millores genètiques en cultius i producció animal, o la recollida de dades en tota mena de processos biològics o biotecnològics per la creació de sistemes intel·ligents que els puguin controlar posteriorment. Bé, l'objectiu d'aquests webinars és, doncs, donar-vos una visió el més àmplia possible de les diverses aplicacions de la bioinformàtica, entesa en aquest sentit més ampli, i l'impacte que hi tenen les noves tecnologies, com els mètodes de tractament de dades massives i la intel·ligència artificial. Bé, ara intentaré compartir-vos una diapositiva per presentar-vos en David. Bé, en David es va llicenciar, suposo que la veieu tots, en David es va llicenciar en Biologia a la Universitat de Barcelona i va fer la tesi també a aquesta universitat sobre la identificació i anàlisi funcional d'una família de transportadors d'aminoàcids involucrats en malalties. Posteriorment, va fer un postdoc de 6 anys en l'anàlisi comparatiu i anotació de genomes eucariotes. El 2006 va ser contractat per la Institució Catalana de Recerca i Estudis Avançats i des d'aleshores dirigeix un grup de recerca al BCC. Actualment treballa en l'anàlisi de variacions del genoma relacionades amb malalties, en presència en diversos projectes internacionals i institucions com ara el BIP. Bé, sense més a dir, David, quan vulguis penso que ja tens control de la pantalla. Quan vulguis pots començar la teva presentació.
El que m'heu... Ara, vale. Anem allà. Ho hem fet abans a l'assaig i ha sortit bé. Ara hauríeu de veure sense pantalla completa, right? No? I ara vaig a pantalla completa. Sí?
Perfecte.
Molt bé. Jo no us veig. Well, I cannot see you, but I hope you stay there. So as Xavi said, I would like to present some snapshots of some projects that we are doing, but in the context of a little bit the story and how we got into this data-driven moment and research. So I am working at the BSc. This is a picture of the BSc, very typical one. I encourage everybody to use the computer and also, or at least visit it, because it's quite interesting. In the BSc, our group is a multidisciplinary group that we are dealing and we are worried on the 3 different fronts or working on 3 different fronts. So all our projects have one question that drives the whole project. So this would be this part up here where we concentrate our efforts in answering biomedical questions, usually the identification of mutations in relation with disease. This is basically what you will see in the talk is the general trend. All the bioinformatics groups know that if they go for a biological answer, they also have to develop or adjust bioinformatic methods and procedures for data analysis. And this is another activity that we do in the group. And finally, the last 6-7 years, we've been involved also in cancer on the development and the design of infrastructures to manage and to analyze large amounts of cancer data. And I will be explaining a little bit of each one of these. So as a general introduction, we know that we aim towards a personalized medicine. We've been talking about personalized medicine for the last 20 years, but still not there. I remember 2006, 2007, everybody was talking about 4P medicine, precision medicine, all the companies. Then nobody is talking anymore, or not so much. There are other trending words, but the point is that personalized medicine is not there yet. Okay, we are not there yet. We are building the bricks. And what we want to get for personalized medicine is precise diagnosis, personalization of the treatment. And one of the promises of personalized medicine was the prediction and prevention of diseases. But this is where we are farther from all the possibilities and all the promises. It's not that we want this now and we didn't want this before. Actually, we always wanted a better medicine. The difference is that now we have the data that allow us to do this much faster, wider, with more detail, so much better. That's why we now include this data-driven world, include the personalized medicine. And this is basically the generation, the management, the standardization, the analysis and interpretation of omics data. I will explain the genomics because genomics is my field. There are other nice developments that you will hear in other webinars of the BIP. They are also interested, but I will concentrate on genetic markers, okay? So mutations that cause diseases. And if we see how medicine now is considering disease in general, and here I'm talking about diseases with a genetic component, which are most of them. If somebody hits you in the head, it's not, but most of the others have a genetic component, small or large, but there is always a genetic component. And here you can see that when you have a mutation, this defect, this mutation propagates inside the body. So this mutation goes into the RNA, the protein becomes a drug target, okay? And then normally the gene, the mutated gene is involved, the function can be involved in different signaling pathways, different metabolic pathways that at the same time can be involved in different cell types, which can be in different organs. And all these mutations can be translated into different symptoms, which makes very difficult to go from the right side of the slide to the left, which is what we want. We want to identify the mutation. We want to go to the origin of the disease, okay? Not only to develop drugs, but also to understand the disease. And this is basically one of the major goals of biomedicine or genomics. But during from the 80s or 90s compared to now, there have been lots of changes, okay? So the way we were looking at genes that cause diseases was always through a mutation or through alteration of the gene. And usually in 1995, where I started my PhD, this work was confined to the molecular biology field or the molecular biology community, okay? So normally you had some candidates, and this is why I said from function to genes. But that time, for example, in my thesis, I functionally characterized some proteins, some amino acid transporters, And then later, I connected with the genetics. I found we connected through some diseases, and then we found some patients, we found some mutations, and then we were able to connect the mutation, that gene, with that disease. But for one or two genes and one or two diseases, it took me 5 years, a lot of energy, a lot of money as well. So it was very efficient because you knew the function of the gene. That's very important. But in terms of discovery, it was quite slow. Okay. Around the year 2000, the technologies for DNA sequencing mostly started to emerge, and new technologies which made the sequencing and also the analysis of genomes much faster, much easier, much cheaper. Together with the developments of automatization of laboratory processes, robotization, and starting to have a deep knowledge or a better knowledge of the biology of the genome, okay, or the annotation of the genome, what is each part of the genome doing, basically, these three elements generated a big change in how we now study diseases, okay? That's why I said here, this is a paradigm shift. With this combination, we can now generate tons of data from different patients around the same disease. And this gives us the possibility of enhancing by a factor of 100, 1,000, what you want, depends on the data, the discovery of markers, okay? What I told you before, that in my thesis was 5 years, one protein, one disease, or two proteins, two diseases. Now, what we can do is to explore widely for one disease, all the genome. So we might end up with many mutations and many candidate genes for that disease. Okay, so this is what it makes a difference now. And with this data, also, it's very important that other communities appear or are enhanced. For example, our group, we could be doing genomics, but towards biomedicine, this has now allow us to go towards biomedicine, because usually genomics was confined or was doing things about evolutionary questions and comparative genomics. But this type of data allow us to play also with biomedicine in genomics. Also, this data brings other communities like more towards engineering or mathematic communities that come with the expertise of AI. Okay, so all this data is not only providing more possibilities, but also is enriching our research environment quite drastically. And again, I'm talking about genomes, but you can apply it and you can see that the burst of data has gone is crazy, and all types of data are being now generated by thousands or by millions. Okay, proteomic data, transcriptomic data, metabolomic data. So this is now quite spread. There's one thing here. There's a pity, and that's the communities that we are doing genomics, maybe transcriptomics, but then we are not doing proteomics, and the communities are different, and there is not much communication among them. But this is something that we have to do that we have to solve, because at the end we want to integrate different data types, okay, to construct better and more complex biomarkers. This is a very schematic slide to show the general approach behind most of the analyses that are data-driven in biomedicine, okay? You first have some features, some data, could be genome, transcriptome, again, all the types of data. And what you want to see is what are the differences, for example, if you use transcriptome, what are the differences in gene expression between the cases, the patients, and the controls? Okay, this is the basic question. And for this, you can use classical statistics, you can use artificial intelligence, machine learning, preferable, but you can also use deep learning, okay? You need some computation. This is where bioinformatics comes, okay? With this, you can do marker discovery, classify patients, this is very important, and at the end, improve clinical protocols, okay? But this is the scheme where most of the type of studies, large-scale data studies, would follow, okay, even AI. If we go to genomics, we can divide the study and the type of diseases based on the type of mutations that are the cause of these diseases. For example, when we are born with a genotype, with a specific genotype, with some germline variations. These germline variations already give us some protection for some diseases. Some people have better immune system, for example, than others, we know. But also, some of these variation can predispose us to disease. So this is not really causing disease, but is pushing you towards the cliff, let's say, but you still need some environmental, smoking, diets, contamination, whatever, and environmental factors to trigger the disease normally, okay? And this disease come late in age, you will see now. If you're very unlucky, you in your genotype, you also can get some rare variants that are causing rare diseases. These variants are variants that are very penetrant, so their effect is quite high. Okay, you normally have the disease from the first day or even before born. You have some defects, okay, because this is, and normally they are as you will see now, affecting proteins quite a lot. On top of this genotype, during your life, other mutations can appear, okay, which would be the somatic mutations, randomly, but because our system does not work perfectly, and also help with the help of smoking and the sun and other mutation agents, you can get somatic variants in some of your cells. This normally you don't transmit this. Okay, no, you don't transmit it unless it goes to germline cells. But in principle, you don't transmit this to your kids. And sometimes, most of the times, these are neutral, but sometimes can cause disease, which are basically the cause of cancer, as you will see now. So with these three types, we can differentiate common complex diseases, rare diseases, and cancer. Not only because the analysis behind are different, also because the type of sequences sometimes is also different. Also, the goals are different, and the communities are different also. Okay, so people working on cancer normally don't know people working on complex diseases, and the other way around. Okay, cancer in this case you can take as a complex disease as well, because you can have predisposition to cancer, BRCA mutations, and so on. But here, cancer, I will only consider it around somatic variation, not germline variation. Okay. So starting with rare diseases, the rare diseases for us are quite boring from the point of view of research, because they're of course very important medically, but from the point of view of research, they are not very challenging, because normally 80% of the times you already find the mutation of a patient that you don't know or you suspect that has some neurological disease or some muscular disease, and then there are some gene panels, or you could go to whole exome, because variants that cause rare diseases, okay, normally affect the gene, normally affect the sequence of the protein. That means that you could find it in an exome. You normally find these mutations in exons, okay? These, even though they are not so exciting from the point of view of research, they have a high applicability into the clinics, because hospitals already have a genetic counseling or genetic diagnosis department. So there was people already doing this in hospitals. So all you had to do is change the technology. Before they were getting the sequences from Sanger, and now they are taking it from gene panels or from exons. Okay, so this has a lot of applicability, and this understanding the genomic part and helps understanding, so helps in the diagnosis, helps understanding the disease. But rare diseases, the point now is to find treatments, okay, which is the bottleneck for rare diseases. Going to complex diseases, this is where we are doing quite a lot of things. These are based on, as I said, on the general variability of germline variants in the population. Okay, we know that we all are different physically. That's caused for some genetic variants, and also we are different not only physically but also in the predisposition of disease. And these variants are also contributing to that. Okay. So if you take two people randomly from a similar population, you get around 3 to 5 million differences between these two persons. Okay, from the 3,000 million nucleotides. Okay, so it's a very small variance. In all the world, there are more than 300 million variants known. Okay, but still is a low fraction that is different among us. Okay, and as I said, these complex diseases, they have a weak functional impact. Normally, you need many variants that push you towards the cliff. Okay, that predispose you to the disease. Okay, you need to merge with lifestyle external factors to come up with a disease, and this comes normally late in life, 50s, 60s. That's why when you get to 50, 60, every generation will be 10 times higher probably, but you have to start getting your regular checks. So because these variants are not so penetrant as the rare disease variants, normally you don't expect these variants to hit directly the protein. You expect these variants to happen somewhere in the genome, other parts in the genome that normally control the expression of the genes. So the effect that these mutations have on complex diseases to the predisposition of disease, again, is not because we change a function like in rare diseases. It's because maybe one gene is a little bit overexpressed or a little bit underexpressed of the average. Okay, and this puts you more towards the disease, maybe. Okay, so this is the type of variance that we are looking in when we do analysis of complex diseases. And also this comes with another problem, which is that all the variants that we find associated to complex diseases, or most of them, are very difficult to assign to a gene. Okay, one variant that is 300 kb apart from a gene, but could be an enhancer that could be affecting that gene, is quite difficult to say this variant belongs to this gene. Okay, so this means that this type of analysis allow us to predict and allow us to do a lot of different things, as I will show you now. But the bottleneck is to understand the function. Okay, through which molecular mechanism this variant is affecting this disease. Okay, so that's why we need QTLs and we need different resources. For many years, we've been doing GWAS on specific diseases, mostly type 2 diabetes. And here there are some examples of things that we've been doing. I will not enter into details. But for example, these two works, last works that we have done, this Tiger data portal, what we do is actually provide information to be able to link the variant that you found in the GWAS with the functional gene. Okay, in this case, it's pancreatic islet, EQTL plus a database. That is very helpful when you have done a GWAS on diabetes normally, because it's pancreatic islet, and you want to know this variant is connected to this gene, for example. Okay, this is one of the examples of our last works. We have been doing also different models of inheritance models in genetics of disease and reviews. And another important work that we have done in this case was the generation of a haplotype map for the Iberian population. This was in collaboration with Rafa Del Cid in GCAT. And here, using this data, we could get generate this panel, okay, this haplotype map or this haplotype panel that is then very useful to be able to do GWAS. I will not enter into details, but it's very useful to be able to do GWAS then on many other diseases. The highlight here is that this haplotype map was particularly focused on structural variation, because usually haplotype maps only consider point mutations and small indels, and we wanted to consider larger variants. Okay. But for many years, we've been working on single cohorts, okay, or a combination of cohorts, but around one disease. But okay, so the point is that when these big, large population cohorts appeared, we can answer more ambitious questions. For example, UK Biobank is one of the reference databases, and I encourage everybody to use it because you can find lots of things there. It is a database that they offer to the community. You have to pay. It's the business model that they have. You have to pay. Sometimes you have to use the Amazon cloud, but these are, well, their conditions. But the thing is that you have genotyping data, now whole genome data, and all these different variables, okay, metabolites, biochemical measures, protein, all the environment and lifestyle. You have electronic health records, socio-demographic data. You have all types of data around the same people. So it's very, very powerful to start asking more complex questions. And GCAT is a smaller version, but it's also local. And we are also using it for this type of analysis. So which are the projects that we have now lately more that keep us more busy? One is the analysis of chromosome X. Okay, chromosome X in complex diseases, all chromosomes have been quite largely analyzed, and you can see here below a summary. This is a very small figure, but each one of these small dots that at the end make a line is one association of one variant with one disease. Okay, and this would represent the whole genome. You can see the chromosomes here in small. So when you see all the variants that are known already, there are tens of thousands known. When you see across all the chromosomes, you see that one chromosome particularly has less than expected based on its size, okay, which is the chromosome X. And this lack of information in chromosome X that you can see here as well, you can see that this is the number of variants, okay, and the number of associations. You can see that the X is here, and it should be actually around close to the 16, depending on what you take, okay? So X chromosome is clearly underrepresented in GWAS. Why? Because usually you need to apply a different protocol, or at least you have to do an extra analysis to analyze the chromosome X. What happens normally is that they set up a big project, they genotype and analyze thousands of people, and when normally you are with time pressure because of the project finishes and you cannot spend the money, or you need to publish, whatever the reason, normally the option is, okay, leave the X and let's do the other chromosomes. So at the end of the day, you have that you have 0.5% of the no associations when you expect around 5% of the no associations based on the size. So what we wanted to do using UK Biobank and dbGaP, we wanted to recover, to analyze the chromosome X in complex diseases, and also to try to understand other points that I will show you. Okay, so we managed to gather data for 800,000 people, which is quite a lot, combining UK Biobank and dbGaP, and we are analyzing more than 3,000 complex traits or diseases. Okay, some are diseases and some other are traits. And here what we're doing, I'll go very fast, but here what we're doing is basically through all the variants of chromosome X and through all these phenotypes, for each one of the variants and each one of the phenotypes, we're asking 3 questions. Okay, you can imagine the number of of calculations that we have to do here. And we're asking 3 questions. First, with male and female together, kind of, it's a meta-analysis, but also with male and female separately, because we also want to know whether some associations are sex-specific or not. And we have some preliminary data. We have to clean up the final numbers, but here the point is to be able to recover these associations, and also try to see whether we find some correlation or some information about full inactivation and gene escaping, which are 2 characteristics of the X chromosome. I will not enter into details, but we can if somebody has curiosity on this, I can expand later. Another project that we have now ongoing in the lab, it's quite ambitious, is the generation of a disease-wide score for disease risk for each person. Okay, so we want for each person to have an estimation of the risk that this person has for many different, in this case, 2,820 ICD-10 diseases that are in UK Biobank. Okay, so for each one of these diseases, we want to find what is the risk score for each one of the individuals. Okay, we start again with UK Biobank. We start again with lots of genetic data plus environmental data. Okay, we have other metabolites, other data. We need to curate the data, which is where most of the time happens in our work, is in the data curation part. We are going to analyze this, and we are going to build these disease risk factors. And these disease risk factors will have the combination of, because up to now, polygenic risk score, you could build polygenic risk score to predict the genetic predisposition of one disease, okay, but it has been never or very rarely combined with the lifestyle and environmental factors of the person. So what we want to do is to build up for each one of the disease types and for each one of the 2,820 diseases in the ICD-10 code that we can analyze in UK Biobank. There are more, but based on the number of individuals, we can analyze 2,820. For each one of them, we want to generate first the genetic score. We want to generate the lifestyle and the environmental score. And here, and from this, we want to add this and generate a final score for each one of diseases, what we call personalized disease-wide risk score. Okay, and this is for each individual. And also, this is very good because you can start applying prevention protocols. You can see the profile of a person and see that this person has strong risk to develop cardiovascular diseases, for example. Okay, and in this strong risk, you can see which are the environmental factors, the risk environmental factors, okay, that you find more associated to the disease. So you can tell the person, stop smoking. Of course, that's anyway, you would get it. But you can actually, with the type of data that we have, you can even say, stop drinking tea. Okay, we have not done the analysis, but we have a granularity of the data. It allows us to go to that level of detail. Okay, and also we will have the protective factors, because there are some factors that we will find that are protective for the disease. We can encourage these protective factors. So at the end, you can personalize these prevention protocols based on your genetics, okay, and based on your lifestyle and your environment. Okay, this is one of the projects that we have online. And also, we have other projects, just a very brief snapshot of other list of projects where we are using XGBoost or machine learning from the genetic and from the clinical side. Okay, I will not enter into details. We are using type 2 diabetes as a pilot disease, but the point is to go big and also apply it to many other diseases. So this was the complex disease. I will explain briefly the cancer part that we have been doing and what are the latest developments there. Here I will not go much in detail, but you know that the tumor is a strange living thing, complex living thing that sometimes appear in our body. It's very complex, okay? But the most important part that we have discovered these last years is the wide diversity at molecular level of different types of cancer, even if they are diagnosed as lung cancer. Okay, you imagine one diagnosis, lung cancer. There are more subtypes, but for the general public, lung cancer is lung cancer. Well, you can have different patients with different mutations that all lead to what is diagnosed as lung cancer. The point is to understand what are the differences between these patients. Okay, so you need to really start using lots of data for this. This is the typical protocol that is used in cancer genomics. Okay, from a patient, you get the normal and the tumor genome samples, you sequence, you analyze it, you discover driver genes normally, and all these are used as markers to develop treatments, to decide for treatments, to diagnose for many clinical actions. Okay, and we started in 2009, 2007, 2009 with the HGCC, mostly with the HGCC project. We started analyzing tumor genomes, but at different levels of complexity. We started with one sample, so assuming that the genome was an homogeneous ball of cells and all the cells are the same, which we knew it was not, but that was the first approximation. And normally this sample came at diagnosis, okay, diagnosis, biopsy, and then we analyzed that. With time, we understood, and the community understood, that we need to follow the progression of the tumor, because this is as important as understanding the origin is to understand how it progresses. So then the longitudinal studies appeared, okay? And now where we are, at the right side of the slide, where we are now is not only doing longitudinal studies, but also accepting and analyzing the intratumor heterogeneity. As I said, the genome, the tumor is not an homogeneous ball of the same cells. It's a complex population of different clones with different properties. Okay, sometimes a subclone is very malignant and the others are not. So this is the type of studies that are now going on in cancer genomics. During many years, we've been involved in many of these projects from the group. We've been doing finding mutations related to cancer at different levels. Our last study, it was quite nice, where we actually collect with Memorial Sloan Kettering in New York and the Charité in Berlin. We collect a study where we found that in neuroblastoma, there are some amplifications of some regions of the gene, and this amplified DNA stays extrachromosomal most of the time, so in the forms of plasmids, and sometimes it does not recirculate, so it's quite complex. But we found that it can reintegrate in the genome and generate more mutations. Okay, so this is the last one that we published in this sense. And that's to finish, I would like to present this other part that we are doing for the last years in the group, which is related to the design and the development of infrastructure for the management, analysis, and sharing, which is a very important concept of oncological data. Okay. What's the motivation for this? If I don't know what I said it, but you could infer from my talk that we came to a data-driven science moment where the more data that you have, the more discoveries that you get. Okay, it should be quite obvious. Okay, so everything is statistical. So the more data, the more power. And the way to get more data is to share the data, to globalize this type of activity, okay? And what is this more data and data sharing allowing? It allows to develop and to enhance the community, the biomedical AI community, which now it's the engineers talking sometimes with medical doctors and trying to find some consensus. I'm not talking only about imaging, which is the most clear example. I'm talking of other more complex approaches. We need this data, this real data, to improve the analytics, to improve the analysis methodology. We need to test and to design the infrastructure. And at the end, we need it because we need more discoveries and more impact in healthcare. Okay, so sharing the data has all the benefits, a few negative points, but not for the patients, for other communities. So what is the situation now? If you are a bioinformatics group, you want to do an analysis on one specific disease, you need to download data. You can do it. You can go to UK Biobank. You can go to different cohorts in DBGAP, in EGA. But what do you find is that first, let's say you are analyzing hepatocarcinoma samples and you are interested in some viral integration of some hepatocarcinomas. You cannot go to a database and ask, well, you can put hepatocarcinomas, you will get some, but it's very difficult to, I don't know, maybe you want some data set that has some mutations in some genes, or you want some data sets that have that the patients have undergone some type of treatments. Okay, you cannot find these things now. It's impossible. What you have to do is go to PubMed and start checking all the publications about hepatocarcinoma and see which data they produced and whether you are lucky that they left this data in EGA or DBGAP database. But most of the times it's very hard, it's very time consuming. So this findable part, okay, is very, very complex. If you happen to locate one of the data sets, another problem comes, okay? First, some genomes have limited access. That means that sometimes you have located in EGA one data set, you want to download it, and then you have to send an email to the owner of that data, to the PI that has submitted this data. And the owner decides. The database plays no role. The owner decides. Based on the paper, because this normally is published, they must give you the data because the data is open or is accessible according to the publication. But in reality, they do whatever they want. Hepatocarcinomas, we asked for 2 laboratories a couple of years ago. From one, we didn't get any answer, and it was published. And from the others, they said that they were still working on those genomes, and we can talk after a couple of years. So the access is not granted. Okay. On top of that, there is no access of clinical data. This could lead us to another talk, another day, but this is a very, very important problem that we have. No access of clinical data. And this is a cultural problem. There is no clinical data because the producers of the data don't want to share the clinical data. They claim it's a legal problem. Sometimes it is, sometimes it's technical, but the major reason not to share the clinical data by medical doctors is because they don't want to. Okay, I want to make this clear. Then also, if you happen to have all these things in favor, you also need to sign a lot of papers because all this data is protected legally, you know, GDPR. So again, the access is also quite complex. I don't want to discourage anybody. We are doing things and there are a lot of things to do here, but there are some things that we have to be working on. And then another thing that interferes with interoperability that is also required for this FAIR, you can see here, findable, accessible, interoperable, reuse, is the FAIR principle that is applied to the data. The interoperable part is also very important. In our case, what it deals is with the different types of analysis that different centers do of the same data, obtaining different results. Okay. At the end of the day, all the data that is produced every day, thousands of genomes, is not fully exploited. We could exploit it much better. Okay. To try to solve some of these problems, not all, but some of these problems, we launched UCAN is a project, a European project that we just closed a few months ago, where our goal was to generate a catalog. And in this case, we went for the findable part. Okay, we wanted to make data findable. Okay, so what we are doing here, we are actually, I'm not going to be to explain this in detail, but we are assuming a federated structure of data. So each data set stays in the hospital or in the environment that has been produced, but one investigator from different parts of the world can ask to a database, to a catalog, for example, can ask, is there any center that has data with this type of gene mutated? Is there any center that has data with patients that undergone this or with that undergone this treatment or that have had this response to the treatment? All these questions you should be able to do through this platform. This is a very extensive work that we are doing. We are using beacons and we are aligned with Global Alliance, but I will not extend here. The last part I want to explain is related to this, to the same scenario, but now we don't go to findability, but to interoperability. In this case, the problem that we found is that different centers that want to join forces together now have usually different analysis pipelines for somatic variant calling. Okay, well, it means that they provide different results. If you generate a catalog, like we said before, but center A gives some variants, center B gives other variants with different qualities and different scopes, the catalog is not really useful. It's useful, but it's very limited. Okay, you want center A and center B to have the same or nearly the same type of variants or type of analysis. Also, you cannot integrate results from different. If center A has produced some results on some leukemia genomes, you cannot merge them and gain power with other leukemia genomes analyzed by other center because the analysis pipelines are different. So what you have to do is normally put them together and reanalyze them together. Okay, like we had to do in TCGA or pan-cancer or is doing in Arago, a central one. And even more worrying is the fact that still whole genome is not in the clinics, but if we do not put a break and we do not find solutions, could be the same patient with the same tumor in hospital A, it's diagnosed in one way and they found some drivers, and in hospital B, they find other drivers. And this would generate discordant diagnosis, which this is really more. The other part is at research side, but this would be a disaster when applied into the clinics. What are the solutions? Well, we have to have center A and B with the same pipeline. So the easiest one would be just tell center A to copy the pipeline of center B or the other way around, and they will have both the same pipeline. This is an ideal solution on the paper, but it's not realistic because data centers or centers that are dealing and analyzing lots of data, they have different pipelines. They have spent a lot of money to generate a pipeline. You cannot just come and say, okay, all you have generated, just remove it and use this one from now on. Okay, and actually, if you want to do it for different projects, you would have to update different pipelines for different projects. So this is realistically is not a solution. The solution we found is OncoLiner is an approach where we try to improve, but mostly harmonize, standardize the calling between two centers based on the quality. Okay. For example, when you get 3 centers, you analyzed on benchmarking data. I will not enter in details, but when you do this test, what we found is pipeline A, B, and C, which come from real centers with real activity in somatic variant calling. When we do the test and analyze it with a benchmarking data set, what we find, for example, is that 30% of the variants found, okay, so between A, B, and C, all the variants, all the mutations that have been found by all three centers, 30% have been missed by at least one. Okay, so there are 30% of discordant variants. This is a huge number, divided into SNVs, indel, and structural variants. But this is a quite high discordant ratio between what it's supposed to be the same analysis. This is translated into also 18% or almost 20% of the genes expected to be found mutated in this benchmarking data set. 20% of them are missing by at least one center. And among these missing ones, 24 are driver genes, which are very important for cancer, and even 5 of them are actionable cancer genes. This would have a quite strong effect in the clinics. Okay, so to solve this, we have generated OncoLiner. I will be very fast, which solves this or tries to solve this. It gives you an assessment, improvement, and a harmonization between centers, so that the program would say, okay, center A, in order to be similar to center B, you have to add this color, for example, or you have to intersect with this color. Okay, all these recommendations we give to the user, and at the end, we hope that 2 centers can improve their calling in absolute terms and also standardize their calling. Okay, when we apply this to our ABC pipelines, we see that even though we still have some variants that are left that are discordant, okay, we lower significantly the amount of discordant variants, the amount of genes that are discordant, and most importantly, we recovered all the actionable genes that were not found before. This is also we are doing on cancer genomics, on synthetic data. We are going for a synthetic cancer genomic twin, what we call, which I will not enter into details because I'm quite late. This is also a slide that wants to represent that for these data efforts, we need to combine and we need to communicate and merge different communities. Many of you know what I'm talking about. And to understand this community and to be able to talk the different languages is key. Okay. And the final slide is to show that we're going towards a more molecular and genomic medicine, more statistical with artificial intelligence that should be trustworthy. We need a lot of real data to learn, okay, artificial intelligence and infrastructure. And most importantly, we need data sharing so that this is globalized, and this is where we will really enhance this type of analysis. And I thank to the people that have done the job. I have not done much. And this is the real people, people that are doing the work that I have explained to you. Thank you very much. I'm happy to take questions.
Thanks a lot, David. Now for the questions. Actually, the way we will proceed is that you can raise your hand if you have a question. We will activate you. That means if you don't want to be seen, just block your screen and allow your microphone. If you prefer to put your question in the chat, that's also a possibility, but we encourage in-voice questions. So while questions appear in the list, I'll start actually asking you something. This is how often, because you've put it very well actually. One thing is the amount of data that there exists, and another thing is the amount of data that is available for actual use. Yeah. So how often do you actually find yourself working with big data and deep learning as opposed to not so big data and not so deep learning?
Well, the situation is terrible. There are so in EGA, for example, so the problem is that with genetic data alone, you can do molecular discoveries, but you can never do clinical discoveries. Because to know if you find a mutation that is very frequent in a type of tumor, you need to then check whether these people with the mutation compared to the ones that don't have the mutation, whether they have some clinical features. That's why having clinical data is key. And there are hundreds of thousands of genomes in EGA without the corresponding clinical data. So this is a data that we have, in most cases, lost already. Okay, with the amount of money that costs, and then with this data that we have to train those models, and we have to try to get those models up and working. Another problem with AI is the missingness and the quality of data. It's a disaster. And actually, this is where I think everybody doing AI on this should be close to medical doctor or people that understand how to clean up and how to infer missing values, because not always you can infer a missing value with the mean of all the rest, because sometimes it's terrible to do this. So you have to really point, but so we could do much more than what we are doing, only because of this.
I have also another question on the environmental factors, actually. How often is this included in databases? You mentioned that in Biobank this is available, and how homogeneous this is in the database. That is it available for all patients? Same data. Same type of data, same type of questions, or this is very diverse, because I guess the usability of the data will be different in one case or the other.
It is very difficult to go from one data set to another, to jump from one data set to another, not only for environmental data, for genomic data, for genetic data, for other types of, it's very difficult, okay? With environmental data, the problem is that for example, you can use a clinical cohort where the cohort has been generated around a disease, okay, and you can have normally clinical and genetic data, not environmental. But these large biobanks, like UK Biobank, that I encourage everybody to use it because it's the best thing that happened to us, to our community in the last years. You have very well annotated many variables. The problem is that this information is not structured. So people in my group know how difficult it is to navigate and to organize and to sort all the UK Biobank data, the phenotypes. We have it. Anybody that wants to collaborate, we have it. Okay. But it has been very painful. Not because the data is not there, and actually, yeah, there are some fields that I don't want to answer or whatever, but the major problem is that this is not structured. So it's very difficult to parse. This is one of the big limitations.
Would things like ChatGPT and so help actually extract data that is text data, for example, from this sort of databases, or not?
Yes, text data, yes, but the fact that you cannot trust on the semantics, on the content of what ChatGPT is saying, so you can, for me, ChatGPT is only a machine that talks like us, but the content you cannot trust. So this is where we are working. We actually are working on transformers actually to see whether it's not only to reproduce the language. You have to reproduce the meaning. You have to analyze the meaning. This is one of the major limitations that we have. We are trying to apply LLM models for genomics, and it's very difficult. It's very difficult.
No raised hands yet that I can see. neither in the chat. Okay, I do actually have a couple more questions. So, yeah, this EU can, yeah, which is actually very nice initiative. And my question though is, what happens to projects like this when the project ends? Because you said that the project ended already.
Nothing. So this is a bigger problem, and it's how normally the Commission, the European Commission, how the European Commission consider the maintenance and the legacy of the projects. For them, their scheme is you have to do some research, you have to have some companies in your project, and these companies are the ones that should be maintaining through economical activity, maintaining what you have generated. This is because they don't give any money. You ask for money. I mean, yes, they give money to EGA, to some platforms that have been funded for many years. But if we now ask for money for a UCAN 2 second part because we want to maintain and upgrade and make it bigger, no, you don't get money for this. So this is a big problem that has no solution. Actually, well, the Commission knows, and they are starting to change a little bit and
yeah, because
put money
at
on
the
this.
end, if there is, I mean, if this doesn't go forward, all this effort that you have made now is basically lost, right?
Well, it goes for a paper for us. Others can reproduce that. The software is open. Companies can download it. But yes, it's not ideal. But that's the only way, through putting into GitHub and just explaining how it works and hoping that somebody that really wants to apply it. Because another problem that we have is that there are no real application scenarios to test this. For that, hospitals and the medical community should be with us, and they are still not with us. They are concerned, but not with us.
Yeah, one thing that I forgot about the environmental data is how big is the impact actually in your analysis? All these lifestyle and environmental factors.
We still don't have numbers.
Disease.
We don't have numbers yet. We are running now those tests. Yes.
Because I assume in this score that you are building, you will be able to extract which is the contribution from the genetics and which one is from.
Yes. Because for each person you have all the genetics and the clinical history plus many interviews with lifestyle things. Yeah, the data will not be 100% accurate, but you can more or less balance this with the amount of data that you have. Okay. So, but these cohorts are population cohorts. So they are volunteers.
Not necessarily, like I assume.
Yes.
Right?
So the thing is that in 500,000 you have lots of diabetic people. You have lots of people with cancer. You have this big number makes it nicer and possible to analyze diseases at large scale.
Okay, David, since I don't see any other questions in the audience or raised hands, then I think we will close the webinar today. I would like to announce that in May, I mean, these webinars will be done monthly. In principle, the third Thursday of every month, although next month for issues with the agenda of the speaker will be on the fourth Thursday. So the next speaker will be on the 23rd of May, Patrick Alloy, and he will be talking about blending biology, chemistry, and artificial intelligence to enable personalized systems pharmacology. It will be another very interesting talk involving quite a lot of machine learning. I sort of would stick to this term machine learning rather than AI in general in our field. So using a lot of machine learning to try and discover new drugs. So, well, yeah, I expect you all to be with us on the 23rd of May. And with that, we finish this webinar. Thanks again, David.
Thank you. Thank you very much.
Bye.